because they are the base of the food web in the ocean, and they are the drivers of biogeochemical cycles. And so we need to understand both how climate is going to impact marine microbes, but also how they in turn might impact the climate system through feedback loops, because they have the potential to change carbon cycling. So at the most fundamental level in my group, we're studying the rate at which inorganic nutrients are converted to organic carbon and back again, these purple arrows. And so we want to understand the controls on these purple arrows, the environmental controls, and how they might shift as we move forward into the future. So these conversions are, of course, driven by marine microbial ecosystems, as shown here in the schematic, with phytoplankton controlling the flux of inorganic nutrients to organic carbon, and heterotrophs controlling the return flux. And so through my work, we're trying to develop an improved mechanistic understanding of these transitions. And we really are zooming down into the microscale level to understand how microbes interact with each other, how microbial communities assemble, and how these microscale processes impact larger scale ecosystem functions, so that we can generate a better predictive understanding of large scale processes such as global carbon cycling. So I want to tell you two stories today, and I decided to tell both of them because there's actually a lot of overlapping techniques and questions that we use for both of these, even though they're on very different topics. And so hopefully you'll see some of these threads occurring as I tell you these two very different stories. So the first is going to focus on organic carbon cycling by marine heterotrophs and how stochastic interactions play a really critical role in the persistence of organic carbon in the ocean. And I want to say up front, I will probably use marine heterotroph and bacteria interchangeably. I'm really talking about single-celled organisms that are consuming organic carbon. And then the second half, I'm going to switch and transition to talking about how these stochastic interactions and fluctuating environments might influence the ability for microbes to adapt. So there's a very large pool of dissolved organic carbon in the ocean. So OM for organic matter, or DOM or DOC. And we're able to quantify that. So this is a really nice visualization from Van Hansel of global distribution of DOC in the ocean. And we see that there are large scale gradients, both vertically and horizontally. In particular, we see this persistence of a fairly large amount of DOC in the deep ocean, about 40 micromolar of carbon, and then higher concentrations in the surface. And the DOC is made up of many different components that are turning over on many different timescales, ranging from hours to centennial timescales. Because it's such a large pool, it's about the equivalent amount of carbon as in the atmosphere. It's really important to understand what's driving this turnover and the rates at which it's cycling. So there's been a bunch of hypotheses proposed over many decades in terms of why we see this accumulation and kind of consistent level of high organic carbon in the ocean. And the first one is what I'm calling the Recalcitrance hypothesis, and that is that there's something inherent about the chemical composition of DOM that makes it resistant to degradation. The second hypothesis is not related to the intrinsic composition of the compounds, but rather the diversity of compounds. And so it's called the dilution hypothesis, and that is that this large pool of DOM is made up of thousands of compounds, and each individual one has been drawn down to a really low concentration that's at or below what we call the subsistence concentration of the consumer. So it's so dilute that the consumers are not able to access it. And when you have enough of these compounds at very low concentrations, we see you can result in a overall bulk high concentration. And there's a third hypothesis, which is these first two have been published on a lot. The third one is less well known, but there's increasing evidence that there's an ecological dependence to DOM consumption, that it occurs as this result of a co-occurrence of specific compounds with specific degraders. And so my postdoc, Emily Zakum, has been working on trying to understand a DOM cycling and how we could better model it in a mechanistic way, and how we could resolve these three different hypotheses to create, we're thinking of it as kind of a unifying hypothesis on DOM control. And so I'm going to kind of give you the punchline for this bit, because hopefully it will help you understand where I'm going when I dive into the model dynamics. But we feel that we can actually understand DOM control in a context that encompasses all of these different hypotheses if we kind of switch how we think about lability and recalcitrance. So instead of thinking about them as an intrinsic property of the compound, they're a property both of the organic matter itself, but also the microbial community and the interactions that occur between them. And by kind of shifting our framing in this way, we're able to develop a mechanistic model that gives us some predictive ability. So to just kind of highlight what I mean by that, right? So the rate at which organic carbon is cycling is due to diverse factors, which are contributing to that rate. So we have an enormous diversity of compounds that make up this DOM pool with variable structures. And they're being degraded by a diverse microbial community that have very different growth strategies, efficiencies, enzyme kinetics, rates of enzyme production. And all of this is occurring in an environment that is very variable and has physical features such as temperature differences and inorganic nutrients, which also play a role in the rate at which DOC can cycle. So to try to capture the diversity of these interactions, we created a stochastic model to try to kind of explore this space. So the model has a supply of a large number of organic matter compounds. So we're representing thousands of different organic matter types, and their supply is supplied at variable rates. And then each of these types are consumed by a bacterial group, a set of bacteria. We've got thousands of different bacterial groups. And we create a random community consumption matrix, which ranges from specialists to generalists. So we basically randomly assign one is going to be able to eat compound 1 and 3 and 20, but maybe compound 5 can only eat compound 30. So we create this random consumption matrix, which determines which compounds are being consumed by which consumers. And then the dynamics of each of the bacterial groups is governed by a really simple, very standard equation up here. I'm not going to show you too many equations today, but I wanted to highlight this because there's not that many parameters. We have a maximum growth rate, an uptake kinetics for that specific compound, a yield. And so for each of these groups, we stochastically assign these parameters, right? So B1 will be assigned a maximum growth rate and affinity for each of its compounds and going down the list through all of the bacterial consumers. So this is a fully stochastic model, which means we have to run it many times. So we regenerate the consumption matrix and also how we assign the parameter values for each of the bacterial groups. And then we run it each time to steady state and look at what emerges in terms of the carbon concentrations and the bacterial abundances.
You want to get a dumpster in Texas?
Did someone have a question? Sorry, I can't see people. So if you have a question, please interrupt. And so what emerged was a bimodal distribution of the organic carbon pools. And so this was a really robust result that emerged from the model. Again, it's a stochastic model, so we need to make sure that the results are robust and we have to run it multiple times. But we see this bimodal distribution where we have a, let me describe the plot. So this is organic carbon concentration that results in the steady state. And then the frequency of the pools that have that. So for example, this concentration was occurring about 30% of the time. What we saw was a fairly small number of pools accumulating to high concentrations and a large number of pools accumulating to low concentrations. And so this is consistent with what we see in the ocean for my colleagues, chemical oceanographers who actually go out and quantify DOM. And we see that these recalcitrant pools like CRAM and polysaccharides make up a large fraction of the DOC pool and are present at fairly high concentrations. And then there's a lot of things that we think of as kind of labile, simple sugars, amino acids that are drawn down to these very low concentrations. And there's a lot of them. This is the kind of dilution hypothesis part of the DOC accumulation. But what was exciting is because we generated this from a numerical model, we could actually dive in and ask why we were getting this bimodal distribution and what determined the bimodality. And so we were able to show that what controlled that, and in particular, where this threshold was. So why did some compounds end up accumulating versus being depleted down to very low concentrations? And this threshold's related to what we're calling the recalcitrance criterion, which is ecosystem specific. And so you can actually solve the model equations to come up with this Q value. And I have the derivation, so if someone wants to stay afterwards, I would love to walk through it with you. But the key is that the model parameters are what are dictating this emergence. We cannot predict whether a compound will accumulate or be depleted based on one parameter alone, but rather the interactions between the parameters, such as the maximum growth rate of the individual consuming, the loss rate of that individual. And there's also actually this impact of the other pools that the individual is able to consume. And so what this suggests is that whether a compound accumulates in the environment is a function both of the compound type, basically how fast can an organism grow on it, what is the efficiency, but also the community that's present there degrading it. So to kind of put this another way, because I know that it's kind of a complicated metric. So if we take each of those organic carbon pools that we're resolving in the model, which is all of these gray dots, and we plot the steady state organic matter concentration versus the calculated Q value from the equation I just showed you, this is what we get. And so you can see this accumulation of lots and lots of pools at very low concentrations. Again, this is on a log scale. And then a smaller number of pools that are accumulating to high concentrations with this very stark threshold, which occurs at a Q value of 1. And so what's interesting about this is that we can predict where each of these points are going to fall based on the values in the model, but that can shift if any of these ecosystem parameters shift. So for example, if growth rates were to increase due to temperature increases, a compound that had previously accumulated in the environment would end up becoming depleted. And because of this rapid, very sharp threshold, you can get quite big changes in terms of the organic carbon accumulation, rather than a linear or more gradual decrease. So this is pretty theoretical. So what does this mean for the kind of real ocean? So we embedded this within an ecosystem model in a water column. So we've got phytoplankton that are producing organic matter, both DOC and POC, and heterotrophs consuming it. We get the kind of classic deep chlorophyll maximum. This is green, the phytoplankton, this is depth here. We get the classic shape of the DOC curves. So this is data in the light gray from bats and huts with high concentrations of DOC at the surface and decreasing at depth. And then the modeled prediction in the black showing the same profile. And so that's in bulk. But in the model, we have many different organic carbon pools. In the ecosystem model, because of computational constraints, we only resolved 25 instead of thousands. But if we look at what these 25 pools are doing, we see the same distribution we saw for the thousands of pools that we did in the stochastic runs, where we see a lot of compounds being depleted down to these low concentrations. So again, this is the dilution hypothesis aspect, that you have a lot of concentrations present at low concentrations, which can result in a standing pool that's fairly large. And then we have a few of these pools that are accumulating throughout the water column. And I apologize, the yellow might be a bit hard to see here. They're accumulating throughout the water column, and they're not turning over very slowly, and they are functionally recalcitrant. But what we found was particularly exciting is that we have these other compounds in the pools of organic matter in the intermediate range, where in the surface, they are functionally recalcitrant. So they are not being drawn down. They're accumulating in the system. But as you move to depth, you end up seeing depletion of those compounds and them being drawn down to the subsistence concentration. And this matches observations that have been seen, for example, by Craig Carlson at bats, where there's this accumulation of organic matter in the surface during the summer. And when you get deep mixing and injection of that organic matter to depth, we see consumption of it by the deep heterotrophic microbes that are living there. So we're thinking of this as a unifying hypothesis. The idea is that we can start to encompass all of these different aspects. There are certain compounds that are inherently recalcitrant. They are hard to degrade, and there's very few things that can degrade them, and they will accumulate in the environment, and it would be very hard to move them over to that labile pool. There is also a lot of compounds that meet this solution hypothesis that accumulate within the ocean. But there's also this ecosystem dependence where compounds can shift from being functionally recalcitrant to functionally labile. And so the hope is that through this model framework, we can allow for predictions and allow for experimentalists and observationalists to go out and try to test some of these hypotheses and test the model, see if some of the predictions from the model hold up, and we can understand how organic carbon accumulation in the ocean might shift in response to environmental factors. I think I already said that bit. So we had started by thinking about DOM, so it's such a large pool. But we've also been thinking a bit about particulate organic matter, which also shows this accumulation, obviously highest concentrations in the surface ocean. And then as it sinks, it's consumed by heterotrophic microbes. So we also have this vertical gradient. We tend to think of particulate organic matter, or so I'm going to interchange POM and POC, as fairly labile. They're from phytoplankton aggregates. I just saw a talk from Phil Boyd, which is an amazing time-lapse video from the deep Atlantic, I believe, where the seafloor became carpeted in green from a phytoplankton bloom that was exported to depth. So organic carbon that in general we think is accessible to microbes. And I remember being really impressed as a grad student when I was learning about the Martin curve, about how little of the carbon made it out of the surface ocean, right? 1%. It depends on where you are, being exported into the deep ocean. But I've been interacting a lot more with physicists and microbiologists, especially through this Simons-funded PRIME collaboration. And when I showed them this data, they had the opposite reaction, which was, why is any of this organic carbon persisting at all at depth? If it's able to be colonized by heterotrophic microbes as it's sinking, we know that the microbes that tend to colonize particles are copiotrophic. They tend to have fairly rapid growth rates. Some observations of kind of 7 per day, even on hard to degrade CRAM, there was an estimate of over 1 per day. Right, so these are microbes that are not turning over very, very slowly, like some of the deep microbes that are free living. How are we getting this persistence of organic matter at depth? So my postdoc Trang Nguyen developed a model of POM degradation that was trying to capture some of the observations that my colleagues who are studying these processes of microfluidics and some of these synthetic beads were seeing in lab. And so we have in the model particulate organic matter, so it's shown here in blue, that's colonized by a primary degrader, which are these little red guys. They secrete enzymes in the model that convert the particle to low molecular weight organic matter, which is what they're actually taking up to grow on. And this low molecular weight organic matter can diffuse away from the particle due to diffusion or also advective loss as the particle is sinking. The bacteria that are colonizing the particle also detach, and we see this and actually have quantification of this in these microfluidic studies. So they leave the particle at a fairly significant rate, and there's obviously also mortality. So the equations for this are actually quite similar to the ones from the model I showed you that Emily developed, the difference being that we've got this conversion term going from particulate organic matter to this intermediate compound that's being taken up. And we have these dynamics of bacteria leaving the particle and also encountering the particle. So motile bacteria that are able to swim and attach to the particle as it's sinking. So the first thing we did was to compare what our very, very simple model did against actual observations occurring at the microscale. So this is coming out of Otto Cordero's lab at MIT. And what they did is they have these synthetic chitin particles, and they did experiments where they varied the amount of the bacterial population at the initial, during the initial conditions. And what they showed was that if there was sufficient cells present, the community was, the organism was able to successfully colonize the particle and degrade it over a pretty short period of time. But when the cells were less abundant, there was not successful colonization of the particles. And again, this is in a very controlled setting in the lab. I'm not expecting to have extinct communities on particles, but what this is really nicely illustrating is what's known as the Ali effect, which is this population dependent growth rates, density dependent growth rates. And so if your population is below some sort of critical threshold, the population struggles to survive and is not able to successfully colonize the particles in this case. And if you're above this critical threshold, you are able to successfully colonize the particle. And so these circles are the data from Ali, which is Otto's postdoc, from the lab experiment, and the red stars are our model simulation showing that we also show this Ali effect, where if you're below a critical threshold, the populations are struggling to grow. And so why are they struggling? Well, they have to overcome this diffusive and effective loss of the monomer that they're growing on. And they also have mortality and detachment that they have to combat in order to overcome this threshold. And so if we're up here, we have rapid growth and rapid POM consumption. And this occurs if there's a lot of cells on the particle initially, if the particle is really labile and easy to degrade, or if there's a lot of encounter with new cells coming onto the particle. And then if we are below the threshold, we have populations that are really struggling. They have a very long lag phase and they're not able to make it to exponential growth, or that takes them a very long time to get into exponential growth. And in the model, we can actually understand what drives this threshold. And again, that's related to these variables that determine the rate of which the polymer is being able to be degraded. So we wanted to understand how these dynamics played out as particles would be sinking through the water column, and whether this would actually make a substantial difference in terms of the rate of degradation. And so we initialized the model at 100 meters and let these particles fall through the water column. And if we vary different model parameters, and these are just a bunch of example runs, and all the parameter values are here mainly for me to be able to answer your questions if you have any. But we are varying particle size, the lability, which is that rate that the enzymes are able to degrade the particle, the initial biomass at formation, the encounter rate with individuals as the particle falls through the water column, and then the growth rates of the individuals. And you can see that we get a full range of behaviors emerging in the model, from populations that are able to grow really rapidly and they consume particles very quickly. And when that occurs, we see that the particles are gone within the upper water column. The depth at which they reach is mainly driven by how big they are, so how fast they're sinking. We also have populations that are really struggling to overcome the losses. The particles are hard to degrade. They don't have a lot of biomass, or they're not encountering individuals as the particles are sinking. And we see that these struggling populations correspond to persisting particles. And then we have these other really interesting dynamics where we get rescued populations. So these populations were initially really struggling to survive, but then they were able to overcome that threshold and grow and consume the particles fully. So we wanted to look at this in terms of the full water column flux. And so we initialized the model using the observed distribution of particle sizes that we see in the field. So this is just an example of a particle size spectra where we see the majority of particles are small and there's very few large particles. And we've run the model with a bunch of different slopes of this. And I'm just going to show you kind of a standard -3 slope. So we initialized the model using this size distribution in a range of different sizes. This results in 69,000 particles. And then similar to what we did for the DOM model, we stochastically assign the properties of each of the particles. So for each of the particle size classes, we have a number of particles in that size class. We stochastically assign the beta value, which is related to lability, how fast it's degraded, the growth rate of the community that's going to be degrading it, what the encounter will be as it falls through the water column, the initial biomass. right? So we're doing these, again, these stochastic simulations. So think about the encounter and the probability of getting these different interactions between types of organic matter and types of degraders. And then we let each of these 69,000 particles fall through the water column and calculate the rate of consumption, and then sum it to get a POM vertical flux. And so what I'm showing here is that aggregated result. So each of the gray lines is actually an individual simulation of 69,000 particles. And then because, again, it's a stochastic simulation, we're running it 1,000 times because we are wanting to make sure that we sample, fully sample the parameter space. And so we get quite a wide range of solutions coming out, with dark blue is the average of all the 1,000 runs. And the circles are the data from Martin, from the original Martin curve paper. And so we can see that these dynamics, oh, sorry, one more I was going to show you. So, and then if we look at the rate of degradation, the attenuation of each of these little gray lines, we can calculate that rate using a standard exponential decay. And so it's typically quantified with this B value, and we can compare that against observed values from the ocean. And you can see that we actually are matching the distribution quite well for the range of different POM flux attenuation profiles that we see in the ocean. That's just emerging from these stochastic interactions between different communities and types of microbes degrading different types of organic matter. The other thing that's coming out of the model, and we're still digging into this, is that we actually see a shift in the community composition with that results from this. So we see a much more diverse community. So this is, each of the colors is a different, essentially bacterial type in the model. And we see that at 150 meters, we have a lot of different types that are growing and consuming the organic matter. And as we move to depth, we see a less diverse population, which is, again, consistent with the few observations that we have of how the particle-associated community shifts with that. So I don't have time to go into all of the details and sensitivity studies and tests that we did with this. But the kind of main takeaways from this study is that the consumption rates that are emerging due to these interactions between the physical and biological parameters. We see this persistence of POM in the model due to suboptimal growth. And so we can get these long tails of the POM curve due to populations that are struggling to overcome loss terms. But also there's a couple of key features that I think are unique that have emerged here, which is that we get labile particles persisting to depth. And that is, again, due to this stochastic interaction between the particle type and the community type. And so sometimes the right community is not there, the right type of degraders, and or not are there in the right abundance. And so these labile particles are able to be exported, especially if they're large and sinking fast. Also really interesting is that our model doesn't predict that we need to have a large shift in the size spectra with depth. We don't need the small particles to all be consumed at the surface and the tail be driven by large particles. And in fact, if you look at the size spectra over depth, it's pretty consistent, which is, again, what we see in observations. Okay, good. It's on time here. So to wrap up this part of the talk, for both the dissolved organic matter and the particulate organic matter, sorry, I should have taken out the D there. We're seeing that we need to think about lability as an ecosystem property, and it's a really critical ecosystem property, and it can shift based on the microbial community, which I don't think is probably new to many of you who might think about these processes, but being able to actually model them is new, and being able to put them into a mechanistic model that matches what we see in the ocean is allowing us to generate hypotheses that I'm hoping colleagues will be able to go out and test explicitly. We also see this threshold behavior of functional recalcitrance, both for the dissolved organic matter and the particulate organic matter. I showed you that with the Q value for the DOM, but for the POM, if you cross that threshold, the critical threshold, the populations are able to grow and consume the particle. And then the last thing that I found also was particularly exciting is that we get these long time scales of bulk DOM and POM turnover that are slow at depth, but by using model parameter values that are fast in accordance with what we know about microbial growth. So we don't have to have our microbes growing at 0.01 per day or some of these really slow rates that other models have been able to needed to use to be able to match observed dynamics. Our microbes are growing really fast, but there's these other interactions that are resulting in the slow turnover times. And so where are we going with this? Well, one of the things that we're doing is trying to think about the fact that in addition to these stochastic interactions that happen between particles or dissolved compounds and bacterial communities, these organisms are also living in a really variable environment. And I think this illustration from Lambert out of the Stocker lab is a really nice one, right? So you've got these coprotrophic bacteria that are motile, that are swimming between particles in the surface ocean in particular. And what that means is that the environment they experience is really variable, and they're fluctuating between feast conditions and famine conditions. And so my postdoc Noel is trying to think about what this means for how we model these organisms and what their growth strategies might be, because there's a real trade-off between being optimal in the present. So if you're either in a feast or a famine, having your growth strategy be optimal for that situation versus being ready for the next state that's coming, whether it's the feast or the famine. And so we're doing we're using proteomic allocation models and some really exciting theory that was developed for E. coli to try to understand how the growth strategies for these copriotrophs in this fluctuating environment might dictate their behavior. Okay. So I'm going to shift gears now and move away from organic carbon cycling by heterotrophs to adaptation. Again, as I said at the beginning, these are two very different areas of research in my group, but hopefully you'll see that there's a lot of similarities. In particular, for both, we're really thinking about the environment that the organism is experiencing in the ocean. It really embracing the fact that the ocean is a really variable and fluctuating environment for these microbes. And the stochastic dynamics play a really important role in dictating the processes that we're thinking about. Okay, so again, we're interested in understanding how microbes and climate interact. And in order to do that, we will also really need to understand how microbes are going to respond to changes in environment. And think about the environment that they're experiencing from the perspective of the microbe. And so to do that, we need to consider environmental variability happening on many different time scales, from really short to long term, from predictable like seasonal dynamics, but also stochastic, like eddies, for example, that an organism might get swept into as visualized here in this NASA simulation. And we also need to make sure we have the right reference frame. And so as planktonic organisms, microbes are at the whim of ocean currents. And so we really need to think about the environment from a Lagrangian perspective as they are getting transported through the ocean system. And so this study that I want to tell you about is thinking about how organisms might be able to adapt to a variable environment. So instead of just shifting from an old ancestral environment to a new environment, they are instead going to experience a lot of variability and potentially experience that new environment several times with varying frequency before the kind of average state that they experience is that kind of quote unquote new environment. Before I do that, I have to do a little bit of primer on terminology. I apologize, but just a couple definitions basically. So when an organism experiences a new environment, there's a bunch of different responses we need to consider. And the first is just kind of typical phenotypic plasticity. So there's a change in traits, but it's not heritable, it's not genetic, it's fully reversible. So that's the kind of fastest timescale response. And then we're very used to hearing about kind of adaptive evolution. So these are mutations, these are changes in the genome. They are heritable and they are fixed because of their genetic mutation. But there's a third kind of what I think about as a tool that organisms have that's kind of intermediate. And that is a trait change that's due to a non-genetic modification, such as an epigenetic mutation. It's heritable, so that differentiates it from the phenotypic plasticity, but is also reversible. so which differentiates it from the genetic. And so for this study, we're going to consider both these typical genetic mutations or modifications, and I'm going to use shorthand. They'll also try to say genetic of HT for high transmission, which means if your parent has it, you have it, versus these epigenetic type modifications, which because they're reversible, if your parent has it, you may or may not have it. You might lose it. And so we wanted to explore how adaptation, by considering both these fast variation, low transmission, and low variation, high transmission events, interacts with a variable environment. And so this is work my postdoc Nathan Walworth did using a standard evolution model, classic evolution model, the Fisher's geometric model, which is a random walk where mutations are stochastically applied to a population, and then we select based on fitness. So the most fit populations and individuals in the populations are more likely to persist. And we've included in this model both of these types of modifications, both the non-genetic and the genetic modifications. And then we coupled this with a high resolution ocean physical model, so the GFDL model, which is run at 0.1 degrees resolution, to try to get a little bit of accuracy in terms of the type of fluctuations that organisms might experience. And this simulation was done at a 2 times CO2. All right, so what are the dynamics that we see in the model? So this is just a really simple case where we are not using the complicated ocean circulation, but rather just having the organism move to a new environment for a period of time and back to the old environment with this fluctuation. So hopefully this looks similar to the particle fluctuation feast famine I just showed you. These are the same types of dynamics that we're thinking about. What happens when an organism experiences a new environment for a period of time, but then has to move back to the old environment. And so what happens in the model? So this is an example of generation time on the x-axis here, interval. Our time scales are done in generations. And the important line to look at here is the fitness. So that's how well the population is doing in the new environment. And so every time it goes into a new environment, the fitness starts to increase due to these beneficial genetic mutations. But when they move out into the old environment, they start to lose those mutations. And so their fitness drops. And you keep doing that until eventually, in this simulation, again, this is stochastic, so we have to do it many times. In interval 60, the population is able to hit upon a really beneficial mutation, and it sweeps through the population and therefore fixes and is not lost once it has been fixed. Right, so that's a genetic sweep. So what emerged in this model when we allowed for 2 types of adaptation, both these non-genetic modifications and genetic modifications, is we saw a trade-off. So the populations that had very short-lived or essentially no epigenetic or these non-genetic modifications, we saw the behavior I just described. So eventually the population was able to find an advantageous mutation and increase the fitness. But the variability between fluctuations was fairly low. And once we added in these persistent non-genetic modifications, we saw much better environmental tracking. So when the population moved to the new environment, they were able to be more fit more quickly. But it came at the trade-off of a longer time to fix a positive genetic modification. So just to kind of show you that more illustratively, this is a whole bunch of different simulations, and then each of these dots has many different iterations due to the stochastic nature of the simulations. And what's on the x-axis here is how persistent are those non-genetic modifications. So if you're over here, they stay around for 50 generations. If you're over here, they're staying around for less, only a handful of generations. And so what we see is that when these non-genetic modifications are persistent, we see that the sweep, and then the y-axis is how quickly the sweeps happen. We see that the sweeps happen more quickly, but they their fitness increase when you go into the new environment is low. So they're not tracking the new environmental change. Whereas when they are less persistent, we see the sweeps take a lot longer to happen, but they're doing a much better job at tracking those environmental fluctuations. So we wanted to understand what this means for the real ocean. So we ran these Lagrangian trajectories. So we seeded the ocean model with basically little rubber duckies, and we followed them around and tracked what the environment was they experienced in terms of fluctuations. And so what we found was that, and then we also did, and then we analyzed it in terms of two different kind of population types. One was a slow grower who was doubling very slowly, and one was a fast grower. In our case, fast was growing at 1 per day. And so what we found when we analyzed these results was that the slow growers perceived a much more variable environment because their generation times were longer. And so along the same trajectory, they would experience more changes in temperature, for example, which is what we're looking at here. And as a result, they in general had fewer sweeps because they didn't stay in the new environment long enough for those sweeps to occur. Whereas the fast growers perceived a more stable environment because they had the shorter generation time, and they therefore were able to adapt much more quickly to warm temperatures and much more sweeps. And so the kind of generalized conclusions that came out of this was that the difference in generation times can actually result in different adaptation dynamics within the same water mass, because individuals that are growing at different rates are experiencing variability in a very different sense. And then organisms that experience a more variable environment, we would anticipate would have faster response times and slower adaptation. So having more of these potential epigenetic type mechanisms and take longer to actually adapt to the new environment because they're seeing a lot of fluctuations. Whereas organisms that see much more stable environments, we would expect to have these slower response times, so less of these epigenetic type modifications, but potentially could adapt faster, have positive genetic mutations fixed in the population more quickly. So this was fun because we were able to take evolution theory and thinking about these stochastic dynamics and fluctuations and combine it with ocean circulation models. But here we really still were relying on fitness as a really abstract concept. And the kind of classic Fisher evolution model that we use just defines fitness as without any actual meaning. You couldn't go out and easily measure it, for example, in the environment. And so what we've been working on now, the paper in revision, sorry, I should have updated that reference, was thinking, we built a model that actually looks at trait adaptation. So again, these are random walks that stochastically occur as the traits are mutated. And evolutionary biologists tend to like to think about adaptation occurring in these fitness landscapes where organisms are trying to walk uphill to get to the best phenotype or genotype. And oftentimes these landscapes are what we call rugged. So there actually might be multiple fitness peaks where there's a maximum peak, but there's other ones that are high, which you could potentially get stuck on through your random walk. And so our trait-based model is allowing us to create, we've developed a trait-based model which allows us to create this fitness landscape using trait data from experiments and allow us to model how these traits might be adapting to changes in environments and how they might move through this trait landscape. And so the punchline here, and I'd love to talk to anyone after who's excited about these type of dynamics, is that the accessible phenotype, so where the population ends up on which of these fitness peaks, is impacted by your starting genotype or your fitness correlation, your trait correlations when you start. And so when we put evolution into ecosystem models, we need to think of not only how the traits are adapting, but actually how the trait correlations are changing, and in order to understand what our resulting phenotypes are going to be. And so I think I'm gonna, I'm gonna just very quickly show you how we're thinking about moving forward, because the dynamics that I've been talking about, they're stochastic, right? As I've tried to emphasize, we need to run the models many times to really be able to understand the dynamics. You can't run a stochastic model just once and be able to make robust predictions. And so that means that they're pretty computationally intensive. In addition, we're trying to think about how to scale from micro scale to large scale, and these scale are really hard to, that's a huge challenge computationally as well. And where I want to get to is trying to understand again how these microbial dynamics are impacting large scale processes. And so we need to be able to go to these large scale models in a computationally feasible way. And so I've been working on a framework that lets us do this, that lets us take into account shifts in environmental conditions, right? So moving from a feast to a famine, or a low nutrient to a high nutrient, for example, maybe a submesoscale eddy that it results in a injection of nutrients into the surface ocean. But that happens in a non-predictable way. So moving from this kind of framework to a kind of stochastic dynamic system, can we do that in a computational way where we can understand these dynamics in a probabilistic framework opposed to a stochastic? And so I have now, I've been working with a mathematician. So we have a framework now that is working. So this is just kind of a teaser. And what I'm showing you here is a really simple proof of concept. These are histograms for a single state variable, which is showed on the x-axis. And what I'm showing you is actually two overlaying histograms. So there's a gray one, which is probably barely visible. And that is the fully stochastic Monte Carlo simulation, where we are doing the stochastic simulation. And then we've got our new framework we call FIND, which is overlaying on top of it. And this is a fraction of the computational cost. And it's predicting, it shows high fidelity with the Monte Carlo simulation. And actually for this initial proof of concept, we have the actual theoretical predictions that my colleague was able to solve for mathematically, which is in the blue lines here. So both are matching the theoretical prediction, which is good. And so I think that this type of framework, which is basically embracing probabilities to get away from the stochastic dynamics, will allow us to scale up these mechanisms. And just to kind of hopefully convince you of this a little bit, we did a simulation for the Hawaii Ocean Time Series where we use this type of framework to capture variability in nutrient injection. And this is the temperature from MODIS and chlorophyll from MODIS versus our simulation in terms of the distribution observed over a 5 by 5 degree region. And the MODIS image, it has about 4,600 pixels per day, whereas our simulation is running with 85 grid cells. And we're able to get a very similar distribution. So I think that we have approaches that will allow us to take some of these stochastic processes and findings and models that I've been talking about today and bring them to the larger scale in a way that's computationally tractable. Right, so that's kind of where we're trying to go, which is what are the decisions that are happening at the scale of cells in communities, and how is that impacting these large scale dynamics? And so with that, we have a couple of minutes for questions. I thank my amazing group, in particular the postdocs whose work I presented today, Trang, Nate, Emily, and Noel. And my funding sources, a lot of this was funded by the Simons Foundation. And with that, I would love to take questions.
Thank you, Naomi. That was an amazing amount of information, and there's so much to think about. No, I loved it. It's like
I should have chosen one, but I was excited about both. So
it's so cool to see, and to see how many concurrent projects you have. Suzanne, go ahead.
Yeah, Naomi, that was a fantastic talk. Your work is so impressive, and really linking those micro scales to the global scales is not trivial, and you're making contributions on the computational side and the theoretical side is really amazing. And so I want to ask you a question that I think about a lot, and I just can't figure out how it works. You've shown that, for example, the particle colonization, there's some facilitation involved. You have these really rare particles, and they sink. And how does this work? How is there a suitable population of bacteria?
Yeah, so I totally, right. So I think what I was excited about is I think we're finally embracing that aspect of it, right? So I think we don't know, but I have two parts of the answer. One is that the initial population that kind of occurs at formation seems to play a pretty big role in the model anyway. So that's a kind of hypothesis that I would love, right? So I'm hoping that this model, by embracing this point, will allow for targeted field experiments, right? So I was like having a conversation with Matt Church, and it was kind of like a while ago, and before we finished this model, I was like, well, I don't quite know what I need you to measure. And he's like, I'm not sure what I should be measuring. And so one of the things that I think this is suggesting is that, so the community that's present at formation, whether that's coming out of a gut or like in the phycosphere as these things aggregate, those play a really important role, because if they're not there, there's not enough encounter. Like you can't get enough. So we're using the equations that Roman has developed in terms of frequency of encounter of motile bacteria. The encounter is important and can rescue populations, but it's not enough to like fully degrade. Like they can't build up enough biomass. And it also has to happen in the surface, because we're applying this kind of exponential decrease in free living with depth. And so by the time you get deeper, there's just no chance. So I think that I'm hoping that this might, yeah, I've also have that same thing, and I'm hoping that this might point in a direction that may get us more data, which might show that some of this is wrong, and we can then revise the model.
Hey, Naomi, this is Bethany. I wanted to jump in just as a follow-on to that, because I'm wondering if quorum sensing is playing a role, and if there's a way to model a quorum sensing function just from lab studies, because it seems like if there's some chemical signaling between bacteria that are, hailing their friends once a few get started, that.
Yeah. So thank you. So we had this model is the simplified version. So we actually started by trying to like put all this fun stuff in there in terms of cooperative behavior on the particles, kind of quorum sensing like dynamics. Also in terms of the detachment, that really fascinates me. So we see in these microfluidic studies that they don't stay around. They leave food on the table, right? So that they're leaving when there's plenty of carbon there. So like, why are they doing that? So we have versions of the model that have all those dynamics in them. It does impact things, but in fact, the simpler equations capture almost all of the behavior because it's really about getting that critical threshold. So, and the quorum sensing and chemotaxing is kind of implicitly in the encounter dynamics and in another part of the growth dynamics without explicitly being there. So the fun thing would be to get more data to be able to validate and put that in explicitly and say, okay, we'll add in as a stochastic choice is whether you can chemotax or whether you do have quorum sensing to increase your growth rate. And so I think that will help us improve the dynamics a lot. And also what I'm really excited about is like this suggests that the same type of particle falling through different oceans would degrade very differently. And so being able to improve predictions about what those differences might be using things like our understanding of who has quorum sensing capabilities versus or chemotaxis, I think would be so exciting.
I have a question, Naomi, about the DOM profiles you were showing. You pointed out the ones that start to become degraded deeper down. Can you talk about why that dynamic happens again?
Yeah, I went through it pretty quickly, so I do apologize. These profiles are super, super cool. Also because we did a bunch of work with the carbon nitrogen cycling of DOM with depth, and these also end up being ones that could potentially coincide with uptake of nitrate, which is again what Danny Sigmund saw at bats. But that's a whole different paper, which I didn't talk about. So what it is that we shift. So this is, again, this is DOC, so this is not syncing. So the production of DOC is happening primarily, the largest production in the model is happening at the surface because that's where you've got all the phytoplankton activity. And what is being produced at depth is some mortality from bacteria in the model produces some, and then particulate particles that are sinking are also producing some, but at a much lower rate, right? So these pools here are getting mixed thoroughly in the model. They're just persisting and getting mixed. So that's what's happening with them. What I just described is why these guys do that kink. And then these ones, is because you end up shifting your bacterial populations in the loss terms in the model.
Okay.
Because you have a higher standing pool of bacteria. So the loss terms are different. And then also the other compounds that are available. So the other cool thing about this model, which I didn't mention, is that we get priming effects here. So if one bacterial group can eat compound A and compound B, if B is present but not A, and then A shows up, it's able to eat A. And so we get this like enhanced consumption of A due to the priming of B in the model.
Huh.
And yeah, so hopefully that answered your question a bit.
Yeah, as I was trying to think, right, what was the shift that was happening? And it sounds like it is a community shift, but driven by the dynamics.
Yeah, so it's the thing is like, it's not one, or it's the community shift is driven by the differences in supply and in loss and in the community.
Yeah.
Like, yeah. So
Cool.
it's like all these things interacting, which is we end up seeing some compounds that fit the recalcitrance, some compounds that fit the dilution, and then others that are fitting these kind of ecosystem dependent.
Okay, if that is all the questions for Naomi, postdocs and grad students, please stay on the call, and thank everyone else for coming to Bio at Noon.
Thanks, Naomi.
Thanks, great to see you.
Thank you. Bye-bye.
Great to see you too.
There we go. I was like, why am I not exiting? And I have to stop sharing. Yeah. So actually, would you guys have rather heard just one of those stories? I was like, I almost like scrapped the second half last night. I was like, I'm trying to put too much in here. I should have just done one of them, but I'm so excited about both of them that I was just like,
I really liked hearing all of them, and now I was kept opening tabs as you were going through of like, oh, I want to go read more about this, because yeah, I don't know, it's different. It's such a fun way to think about the ocean, and so different from the way I think about it, that I like seeing both ways. Yeah.
I would not have been offended if you were like, yeah, that was a lot. I would have rather gone one. But yeah. I feel like in